to nail down a lot of these high yield facts, and we'll keep kind of adding layers to it as we go. All right, so when we're talking about innate immunity, we're talking about non-specific responses, right? Think of this as like, this is your standard defense. It's not really specific to a pathogen, right? It's just in general, these are our standard defense mechanisms. And I just want to say here with innate immunity, we're not just talking about cells in the bloodstream, for example. I mean, there's a lot of things, a lot of different defense mechanisms that your body has. For the skin, for example, right? The skin is protecting us from interaction with bacteria that are on the surface of, things that we're using every day, right? So our skin is going to be a defense mechanism. The gastric acid in the lumen of the stomach, that's a defense mechanism. So we have a lot of different defense mechanisms. When we're talking about questions in the immunology section, though, we're really going to focus on the specifics here, particularly in the blood and in the tissue parenchyma. So like I said, for innate immunity, it's just like your standard defense you're mounting. Again, non-specific, it's very rapid, right? As soon as you're exposed to a pathogen, you're going to have a rapid response. This is compared to adaptive immunity, right? In adaptive immunity, it takes time to build up that memory response for future exposures. Every response should be equal to the initial response. You're not going to get any memory here with innate immunity, okay? So that's important. Again, no memory with innate immunity, and all of this is juxtaposed, again, to adaptive immunity. Now, when you're talking about, okay, well, what kind of things would set off this innate immunity? What are some of the classic examples here? Okay, so let's just say that we have some immune cell here, okay? So this is going to be our immune cell, and this immune cell is going to, at some point, interact with this bacteria. Now, let's just say, for example, that this bacteria is a gram-negative bacteria. Maybe it's a gram-negative rod, for example. Now, one of the features of gram-negative rods is that they have lipopolysaccharide, right? They'll have LPS. Okay, so let's just say this has some LPS here. And so that LPS is an example of what we'll see as some kind of just general molecule that our body will recognize, right? Some type of pattern our body recognizes and says, hey, this is not good. This isn't something that is not supposed to be in the blood right now, right? It doesn't matter what type of bacteria it is. If it has LPS, the cells can recognize it. And that LPS is an example of one of these pathogen-associated molecular patterns. Now, how do we actually recognize these patterns? Well, our cells, right, our immune cells will have on them receptors called pathogen recognition receptor. The PRRs, the pathogen recognition receptors, are going to look for things like LPS, right? And this is being the pathogen-associated molecular pattern. It's looking for some non-specific pattern that, in general, the body knows this is not good. It doesn't tell you that it's a specific antigen with a specific epitope. It just recognizes things like LPS. And an example of a pathogen recognition receptor, the classic one to remember, is going to be the toll-like receptors, okay, the TLRs, toll-like receptors. And we have that written here, toll-like receptors. So the toll-like receptors can recognize things like LPS, for example. And so these pathogen recognition receptors can bind to these molecular patterns that some of these pathogens have, the classic one being LPS. And when they do that, they'll recognize that this is something that is not right. And so this is going to set off the alarm bells, right? And so this is going to generate some type of immune response, right? We're going to release things like interleukin-1, which we'll talk about, TNF-alpha, right? We're just going to generate some kind of inflammatory immune response. Okay, that's the key. Another one here would be interleukin-8. Okay, so this is going to generate some acute inflammatory response. Notice what I said. This was non-specific, okay? It wasn't specific to one bacteria versus another. It's any bacteria that has lipopolysaccharide in their wall. It's rapid. As soon as it binds, we set off an acute inflammatory cascade. It's always going to be pretty much equal. It's always the same, right? It's not going to be different if I'm exposed 1 month versus another, okay? It's always going to be the same. There's no memory to these receptors. We're just identifying a pattern that we know shouldn't be there potentially. Now, how do we go from this immune cell to releasing all of these different cytokines in the blood? There's one particular transcription factor out here in the cytoplasm. So we have NFKB. Okay, so NFKB is going to go from the cytoplasm into the nucleus, and that's going to code for all of these kind of different acute inflammatory reactants. Okay, so that's how this whole process happens. So just to recap, right? So our immune cells in our blood, they have some non-specific pathogen recognition receptors. The classic one here is the toll-like receptor. There's different variants of these, but these pathogen recognition receptors are going to identify pathogen-associated molecular patterns like LPS, like bacterial flagellin is actually another very classic one. Viral nucleic acids, these are all very classic. So they're going to recognize these. And so these immune cells will then bind, using these receptors, bind to these patterns, and then that's going to cause NFKB, this transcription factor, to go into the nucleus and then encode for these acute inflammatory reactants. And we'll go through all of these in the upcoming videos. Interleukin-1, TNF-alpha, interleukin-8 are all very classic. And these are the things that help stimulate the body to reset the hypothalamus set point, right? So you end up having a fever, and we're generating an inflammatory response to kind of attack this infection. Again, very nonspecific, very rapid. Now, in the category of innate immunity, there's some different things that you want to be familiar with. How do we actually go about targeting this bacteria for destruction. What can we do to actually destroy this bacteria once we've recognized it? Well, the big thing here, in our bloodstream, we have IgM. Now remember, IgM is an immunoglobulin. So IgM will actually bind specifically to its epitope. Okay, so we'll talk about this in the adaptive immunity section. However, IgM in general might just be floating around in the blood. IgM is going to kind of be like your standard immunoglobulin. So I want you to kind of think about it like your standard immunoglobulin. And the one thing that makes this different from some of these other things on here is that it actually is specific to one pathogen. Okay, so immunoglobulins in general are specific to a pathogen. However, IgM isn't involved in memory responses per se. It's just kind of your standard immunoglobulin that's going to be released usually in an initial infection. Okay, and then later on we'll have class switching. Right, when we have class switching, we can upgrade this to IgG, IgE, IgA, right? We'll talk about all of this in the next video. When IgM binds, it does bind specifically, so just keep that in mind. That's kind of tricky, but it's sometimes categorized here in the innate immune response because it doesn't involve class switching and having any type of memory response per se. Okay, the other things on here, complement membrane attack complex, C3b, we'll talk all about this. Lysozyme, which can change the pH of some of these cells for destruction. Lactoferrin, also very classic. So these are just things to be familiar with, part of the innate immune response. And then the respiratory burst pathway, which we'll talk about in great detail in a video to come up on neutrophils. By the way, this thing that we're looking at here, this is going to be a neutrophil. Okay, so we're looking at a neutrophil, and I'll talk about how to distinguish these in just a second. But this neutrophil, how is this neutrophil going to attack this bacteria with this innate immune response using some of these things we talked about? So the first thing this neutrophil can do is it can use phagocytosis. Okay, so thinking of the Pac-Man, right? This neutrophil can eat up this bacteria, and once it eats up this bacteria, it can take the components of that bacteria and put that into phagolysosomes. Okay, so it can put it into phagolysosomes, and then it can use chemicals like lysozyme to try to break down the components of this bacteria, right, as it's digesting it. The other thing that it can do is it can use the oxidative burst pathway, which is what we were just talking about. So it can use the respiratory burst pathway to generate things like superoxide radicals, hydrogen peroxide, to essentially break down the components of the bacteria. So the key is with neutrophils, we have phagocytosis, right? They can use chemicals like lysozyme, for example, and they can also use the respiratory burst pathway, oxidative burst, to help break down these bacteria components. Now, the key thing that you want to remember, though, with neutrophils do not present these bacterial antigens to other cells. That's important. Okay, and that's going to be different from some of these other cells down here, which we'll talk about in just a second. So in general, when my neutrophil in the blood runs into this bacteria, that neutrophil can engulf it. It can try to digest it using phagolysosomes and the respiratory burst pathway. As this is happening, right, this acute inflammatory response occurring, the bone marrow is going to increase the number of neutrophils. It's going to say, hey, we need more of these neutrophils. We need more of these guys so they can go eat more bacteria. The bone marrow just starts pumping out neutrophils. And in the process of doing that, it's also going to release some immature neutrophils. And these are sometimes called band cells. Okay, so these immature neutrophils are called band cells. And I'll talk about this a lot in the hemonc section. But this is why if a patient has an infection or bacteremia, for example, you can use criteria like the amount of band cells that the patient has, or the percent band cells, to help give you a higher conviction that the patient has an infection. Remember, we talked about SIRS criteria, right? Having a high percentage of band cells would suggest an infection. And so that's what's happening. We're pumping out more of our neutrophils, as well as some of the other cells here, like the eosinophils, for example. So this is an example of an eosinophil. So notice the eosinophils, they're going to stain pink. Okay, so eosinophils in general stain pink. Again, we have the multi-lobed nucleus here with eosinophils, multi-lobed nucleus here with neutrophils. And that's kind of why the neutrophils, eosinophils, basophils are all classified as PMNs. The P is polymorphonuclear, right? Polymorphonuclear. They're going to have multiple lobes in their nuclei, right? And so that's what we see. Again, eosinophils staining pink, and you can see the neutrophils staining a little bit lighter blue in their cytoplasm. And then here we have a basophil. Okay, so I'll do this one in blue. Okay, so the basophil, you can see this one's staining a little bit deeper blue. And just to keep things simple, I don't want to go into too much detail on this here, but the basophil, like the eosinophil, they're both seen in parasitic infections. The basophil more classic for allergic reactions overall, and the eosinophil more classic for parasitic infections. But you can see basophils with parasitic infections as well. The thing about basophils basophils sometimes gets overlooked. They're non-phagocytic, so they're not going to be digesting bacteria. Eosinophils, on the other hand, can do that. And the mast cells we sometimes see with allergic reactions, mast cells usually will not have multiple lobes to their nucleus, like you see here with the basophil, like you see here with the eosinophil. Mast cells are not included in that PMN category. So all of these cells are going to be getting pumped out usually when we have some kind of inflammatory cascade, like we talked about. All started up here when that immune cell used its pathogen recognition receptor to run into, in this case, LPS, being one of the pathogen-associated molecular patterns, causing interleukin-1, TNF-alpha, interleukin-8 to get released because NFKB, the transcription factor, coded for them as this interaction took place between the pathogen recognition receptor and the PAMP. All right, just a couple other things here. So the monocyte, so this cell is a monocyte. Okay, so the monocytes sometimes can be tricky to differentiate from some of these other cells. So the monocytes usually have more of a horseshoe kind of appearance to their nuclei. That's classically how it's described. Now the monocytes, remember, the monocytes are primarily going to be in the bloodstream. Once these monocytes get into tissues, when they migrate into tissues, they're going to become macrophages. And those macrophages, just like the neutrophil, they can go into that tissue and they can use phagocytosis to try to digest the bacteria. And the difference between the macrophages and the neutrophils is the macrophages actually can take the antigens from the bacteria and put them on the cell surface to present them to other cells. So we'll see that in the next slide. Now, monocytes don't just turn into macrophages, they can turn into dendritic cells, which are also antigen-presenting cells. So just keep that in mind. When we talk about antigen-presenting cells, the dendritic cell is kind of like the poster child for that whole process. So remember, dendritic cells can come from monocytes as well. Then we also have things like natural killer cells, which I'll talk a lot more about when we start to talk about major histocompatibility complex and that type of thing. So we won't go into too much detail on that here. But again, all of this is part of your innate response and checks a lot of the boxes that you see up here. So just to recap this, the primary function here is rapid. That's important. It's a very rapid recognition and hopefully a resolution of some new infectious agent. Now, when I say new here, it doesn't mean that we weren't previously exposed to that infectious agent. It just means that we're now exposed to that infectious agent. It's a new infectious agent. It hasn't been around for a long time, right? It's not a chronic infection. It's something that we were just exposed to and now rapidly recognizing it and then attempting resolution of that infection. Particularly effective versus fungi and parasites. And we'll talk more about why that is as we proceed here. That's not to say that innate immunity doesn't work against bacteria or viruses. It just isn't as effective. And the last thing here is that this is all germline encoded. So what are we talking about germline encoded? That means that the innate immune response can vary from one person to the next person. But the key is that this whole process in the innate immune system is not going to change over time. Once you have your genetics for innate immunity, that's it. That's what you got. Okay, it's not going to get better. It's not going to get worse. I mean, over time, right, as we age, our ability to respond to infections kind of comes down a little bit for other reasons. But in general, we're not going to generate more memory responses or anything like that. Remember, this is no memory response. It's equal to the initial response. It's always going to be the same. So whatever you have that's germline encoded, that's what you're going to have pretty much for the rest of your life. And so this all develops in utero. When we're still a fetus is when this whole process is going to form, which is going to be different from what we talk about in the adaptive immune system in just a minute. So this is all inherited from mom and dad. And the last thing I just want to touch on here, I kind of brushed over this, but really quickly, we talk about these neutrophils as polymorphonuclear cells. Here you can see that this neutrophil has 3 lobes. And sometimes people say, well, how do I tell a neutrophil from a monocyte? Right? So the neutrophils, like we said, are going to usually have multiple lobes. Classically, neutrophils will have 3 to 4 lobes. Most of the time, they're going to have 3. If you see a cell that looks like a neutrophil, but it has like 6 lobes, remember, that's a very classic for megaloblastic anemia. Okay, so think about folic acid deficiency, B12 deficiency. I just want to remind you of that. I think I wanted to talk about that, but I forgot to bring it up. But that's particularly important when you're looking at these cells because you have to distinguish these. Remember, eosinophils stain pink, basophils are going to stain darker blue, mast cells will not be PMNs, neutrophils with more than 5 to 6 lobes, now you're starting to think about B12 deficiency, monocytes classically with more of the horseshoe appearance, and then it's going to usually have a higher nuclear to cytoplasm ratio. Okay, so just keep all of that in mind when you're looking at these cells. We're going to talk about them more in HEMONC, but I just wanted to cover that here. So now that we have some basis on innate immunity, let's talk about adaptive immunity. And what I'm going to do is I'm going to go through this slide pretty quick, and then we're going to go into how this whole process works, and then I think it'll make a little bit more sense. So I'm just going to kind of hit the points really fast on this slide, We'll circle back though. Okay, so again, this is going to be a specific process. Okay, so we're targeting a specific antigen, a specific epitope usually, or a specific group of epitopes potentially. The speed in general, when we're talking about adaptive, it's going to take us time to adapt to that response. However, if we're exposed to that pathogen in the future, we have a heightened response from previous, and that's because we have some memory that's present after the exposure. Again, there's some previous epitope that we were exposed to that we're targeting. That epitope bound to IgM initially, and eventually we'll have class switching, which we'll go through here. And the key points here to remember, some of the key players, at least T cells, CD8 and CD4, we'll talk about that in a minute. B cells that can turn into these antibody producing factories, plasma cells, specific immunoglobulins after class switching. So these are all part of the adaptive immune response. And again, I'm going to go through all this in a second. I'm just kind of introducing it here. So the primary function here, again, is going to be recognition and resolution of a previously encountered infectious agent. For example, people that may have been exposed to COVID-19, right? They may have developed some memory response to future exposures. But then when you introduce new things like a Delta variant, for example, that might change the body's ability to respond to that pathogen, because now maybe it has some protection against that epitope that we were using to mount an immune response against. So we'll get into the details on that. It's going to involve more of VDJ recombination and hypervariation, as opposed to having something that's germline encoded. With VDJ recombination, which we'll go through this all in the antibody video, we're generating different receptors that can hopefully target that epitope that we're being exposed to. Okay, and so the key here is these are not germline encoded. They don't come from mom and dad, right? It's not like the innate immune system. These are all going to come from our initial processes of VDJ recombination when we're creating the variation in our receptors to have something that hopefully can bind to some of these epitopes. Let's go through what this process of adaptive immunity looks like. And this is really going to be the first layer into these immuno videos that kind of put the big picture together on what's happening here, because it's really easy to get lost in the weeds on some of this stuff. So the first thing is, like I said, dendritic cells are going to be our poster child for antigen presentation. All right, so let's see how this process starts off. So we're going to go back to our example in the last video. We had some bacteria potentially that came into the bloodstream, and we talked about what the neutrophil can do in the macrophage. We can digest it, we can have it go through phagolysosomes, and we can even potentially present that antigen. And so that's what we're going to talk about here. How do we present that antigen? So again, dendritic cell here is going to run into this pathogen. Okay, so it's going to run into this pathogen, and the dendritic cell, what it can do is it can try to digest that pathogen. So again, we're just going to pretend that we're undergoing phagocytosis here, right? We're going to eat this pathogen, we're going to digest it, and then what this dendritic cell will do after it digests the pathogen, it can present components of that pathogen on its surface. Okay, so it can present components of this pathogen. Now remember what we just said, neutrophils do not undergo antigen presentation. It's important to know what cells actually do undergo antigen presentation. Again, the classic one is dendritic cells, that's why I'm using it here, but this could be a macrophage in a tissue. It could be a monocyte in the blood. It can even be a B cell, which we'll talk about in just a second. But for the example right now, let's stick with a dendritic cell. So again, it's loading these protein components, right? These are proteins from this pathogen that it digested onto its cell surface. Now we're going to start to integrate some of the material from the last video. So let's just say we have a lymph node here. Okay, so we're going to say this is a lymph node. So this dendritic cell can travel to a lymph node now with these antigens presented. And inside of our lymph node, remember what we talked about. In the paracortex, right? In the paracortex, we're going to have some T cells here. Okay, so we're going to have some T cells. And this dendritic cell is going to come in and it's going to present those antigens to the T cell. And so the T cells in here, there's going to be a whole bunch of different T cells. A rare number of these T cells might actually have a receptor that is able to bind specifically to this antigen, okay, to the epitope of this antigen. And this is where that hypervariation, that VDJ recombination comes into play, because you have to have a whole bunch of different receptors to have one that eventually binds to this very specific antigen. And so let's just say that this is the T cell, in the paracortex that binds specifically to this antigen. So this T cell is going to wake up, right? It bound to this antigen, and now this T cell goes from being naive meaning it was kind of just sitting there in the lymph node, not really doing much, to all of a sudden it found its antigen. Now we wake it up, right? So I'm highlighting in yellow here. Now it's activated. That's what we would say. It went from naive to being activated. Now we have an activated T cell. Now remember, this thing here in purple, that's the T cell receptor. Okay, so the T cell receptor bound to the antigen, but what exactly is that antigen on the dendritic cell? Well, the dendritic cell uses the major histocompatibility complex to present the antigen. So it uses a major histocompatibility complex, an MHC. Okay, now, dendritic cells can use different types of MHCs. Okay, so it can load the protein components onto an MHC receptor 1 or an MHC receptor 2, and there's a consequence to this. And I'll talk about that in just a second, but just remember, it can load it on MHC 1 or it can load it on MHC 2, and depending on which MHC it's on, it's going to target different T cells, and we'll talk about that in a second. All right, so this is the general process. We digested a pathogen, we took the components of that pathogen, we put it on our major histocompatibility complexes on our dendritic cell, went to a lymph node, presented it to a T cell that has that same type of receptor and activated that T cell. Now, I also told you that interestingly enough, the B cells can also present antigens. So down here, I have my B cell. So what happens if my B cell runs into this pathogen? Well, the B cell can also phagocytose this antigen. Okay, so B cells are also antigen-presenting cells. They can use phagocytosis, break up this pathogen, and then present the pathogen on MHC complexes. Okay, so the B cells will also have these MHC complexes that they can use to present the antigen, kind of like the dendritic cell. The B cell can also go travel to the lymph node, right? It can go travel to the lymph node and it can find a T cell, just like the dendritic cell. And it can also, just like the dendritic cell, present this antigen to the T cell. Okay, so again, when the B cell goes to the lymph node, right, it's presenting the antigen on MHC2 to the T cell receptor on the T cell that matches that antigen, and then our T cell wakes up. T cell is now activated. Now, what's the difference between these two? Okay, so let's talk about that. So when the B cell presents this antigen, remember, this T cell is going to get activated, and there's a specific connection that's going to happen between the B and T cell. And I'm going to talk about this more as we kind of move through the series here. So if you're a little overwhelmed, don't be overwhelmed by this yet, but this is going to be the CD40 ligand. Okay, so this is kind of like a co-receptor that's binding as the B cell binds to the T cell. And that CD40 ligand binding is going to allow the T cell to send a signal to the B cell and say, hey, I need you to undergo some class switching. So this additional binding is going to have the T cell telling the B cell to potentially undergo class switching. So what happens is the T cell is going to send a signal to the B cell, right? And now the B cell, okay, can turn into a plasma cell, and then it can just start pumping out antibodies. Okay, and again, and if we undergo class switching, we might start pumping out antibodies like IgG, IgE, IgA. It's all going to depend on what type of cytokines that T cell releases to tell the B cell, hey, this is the antibody I need you to produce. I need you to class switch the heavy chain on IgM to IgG, IgE, IgA, all from this process where the B cell presented the antigen to the T cell. Okay, and like I said, we're going to go into that in even more detail. I'm just kind of laying the framework because this is kind of a complicated process. Okay, but essentially, again, when the B cell binds here to the T cell, and we have CD40 ligand binding, the T cell will stimulate the B cell to become a plasma cell, which is essentially like an antibody producing factory, right, to produce these antibodies. Okay, and we can also have the class switching because of the CD40 ligand binding. That's the important part to remember. And this whole thing where we have all these antibodies being produced, this is known as humoral immunity. When you actually class switch to IgG or IgA or IgE, that takes a while. That could take a few weeks potentially. And that's why we say, adaptive immunity in general, adapting these memory immunoglobulins. IgG, you think of more as your memory immunoglobulin. It shows that you were previously exposed to that pathogen. When you're looking at hepatitis, for example, very frequently we're going to look at the IgG. Again, it's going to differ depending on the type of hepatitis. But IgG is going to tell us we were previously exposed or we were vaccinated because at some point we had exposure to that specific epitope or antigen. Whereas IgM, again, is going to be kind of more of a first line antibody or immunoglobulin. Okay, so IgG is part of the adaptive immune response. It takes class switching to get IgG. You have to have a B cell binding to a T cell, activating that T cell, right, stimulating cytokines to cause the class switching to IgG. Now, the thing to remember is that we bound to a T cell here to cause this whole process to happen. But remember, this was all on MHC2 in B cells. Okay, so the way to remember this is that you always want to get to 8. It's kind of like the rule of 8s here. So if we have anything on an MHC2 receptor, if we multiply 2 by 4, we get to 8, which means the reason I'm showing you this is because MHC2 receptors will bind to CD4 positive T cells. Okay, so CD4 positive T cells are kind of like your helper T cells. Okay, so the helper T cell, so this in this case would have been a CD4 positive T cell because the B cell loaded it on MHC2. And we want helper T cells to release cytokines to help stimulate these B cells to turn into plasma factories. Okay, so that's how you remember. If anything is loaded on MHC2, it's going to bind to CD4 positive T cells. Those are the helper T cells. Now, conversely, with our rule of 8s, if we load something on MHC1, right, 1 times 8 equals 8. So this cell up here, let's say that we loaded it on MHC1 on our dendritic cell. If we loaded it on MHC1, then it would have had to bind to a CD8 positive T cell. CD8 positive T cells are going to be your killer cells, okay, your cytotoxic T cells. And so these cells, what this CD8 positive T cell will do is it will actually release enzymes to destroy that dendritic cell. You might be saying, well, why is it destroying it? Remember, the dendritic cell is infected with something. CD8 positive T cells usually will be very effective when we're talking about viruses. Most of your cells should express MHC1. So if a T cell sees that another cell is expressing an antigen on MHC1 that's not supposed to be there, it thinks that that cell is infected. If you have a virus, that's exactly what happens. If a virus infects a cell, the T cell will recognize it on MHC1 and it will kill that infected cell. So the cell in some ways is essentially sacrificing itself knowing that it's infected, right? Because a virus can hijack a cell and then use that cell's machinery to make more viruses. And so the idea here is the CD8 positive T cell saying, hey, you're infected, you might be infected with the virus, let me wipe you out so that this virus doesn't reproduce even more. Okay, so that's like a very simple explanation to it, but that's essentially what's happening. And that's cell-mediated immunity. The T cell is directly targeting the infected cell. So again, CD4 positive T cells are going to bind to MHC2, which are very classic for the antigen presenting cells. In general, when these dendritic cells are presenting things to the T cells, usually we'll talk about it with MHC2. The tricky thing here that I want you to remember though, and again, we're going to keep talking about this, is that in general, the cells that are going to be the antigen presenting cells, they're pretty much going to express MHC2. So this dendritic cell, it could express this pathogen on MHC1, it could express it on MHC2, for example. If it expresses it on MHC2, it would be binding to CD4 positive cells. So this dendritic cell can bind to CD4 positive cells or CD8 positive cells because it can load its protein components on MHC1 or 2. Almost all of your cells are going to express MHC1. Let's say it's not an antigen presenting cell. It's kind of just like your everyday cell in the body gets infected with a virus. What happens is that T cell, CD8 positive T cell, if it runs into that cell, it's going to kill it, right? It's going to destroy it because it doesn't want that virus to reproduce. Okay, so when you think about MHC1 and CD8 positive T cells, think about kind of like your everyday cells expressing MHC1. Of course, dendritic cells also will express MHC1, but classically we think about dendritic cells usually with MHC2. So I don't want this image to confuse you too much. So just to recap this, going back to the innate immunity, this pathogen enters our body. We have a bacteria enters our body, okay? The bacteria can run into a neutrophil, for example. The neutrophil might digest it, and then the neutrophil will try to break it down using the phagolysosomes, using respiratory burst pathway, and it's going to do all of that because it used a pathogen recognition receptor to recognize LPS, for example, on the surface of this pathogen. Okay, it might use toll-like receptors, right? Or it might recognize the bacterial flagellum. It'll recognize some pathogen-associated molecular pattern, right? That's what we talked about before. Using pathogen recognition receptors, very nonspecific, generates an immediate response. We have interleukin 1, interleukin 8, TNF-alpha being released for an acute inflammatory response. That's what happens initially. Now, eventually, we might have some antigen-presenting cells that are going to run into this pathogen, like this dendritic cell, and it'll digest this pathogen. It'll present that pathogen on its MHC2 or MHC1. It could be either one. It'll go to the lymph node, present that to a T cell, activate the T cell that binds specifically to that antigen, right? And then that T cell can either destroy the dendritic cell if it's on MHC1, or the T cell can release cytokines if it's CD4 positive. Okay, B cells can do the same thing. B cells can run into this pathogen. They can present this pathogen to CD4 positive T cells on MHC2, and that can cause class switching. We can turn these B cells essentially into plasma cell factories, releasing some of these very specific markers that we talked about in the adaptive immune response. Okay, IgG being very classic to show that there's been some previous exposure because we had to have class switching prior. And remember, if we go back to the first video, as these B cells are pumping out these IgGs, IgEs, IgAs, remember, they're all going to happen in the germinal center of the lymph node, right? So you're going to get that pale staining in this area after these B cells have started to go through class switching and are starting to kind of replicate themselves. Just remember, the majority of these B cells might go to be plasma cells, but there'll be a small number that kind of hang around in the lymph node and become memory cells. Okay, and that's true for the T cells too. There'll be some memory cells that stick around. Okay, and so those memory cells, if the exposure happens again, if we run into this pathogen in the future, right, maybe 2 or 3 years down the road, we get infected with that same pathogen, these cells are going to get this epitope presented to them, and they're going to generate a very strong response very fast. That's going to involve antibodies like IgG from the class switching, for example, before, and we'll be able to readily target this pathogen with a much more robust response. In some cases, not even getting to the point where we have a huge acute inflammatory reaction because we're able to respond so quickly. So the patients might not even have symptoms when they're re-exposed because we can target the pathogen much more rapidly and effectively. And like I said, if you're a little overwhelmed by this, we're going to go through this piece by piece through this video series. So don't worry too much about this, but I at least like to introduce it all here, and then we'll kind of build on it as we go. Pause the video if you need to. We'll give you 15 seconds to read the question, and then we'll go through the explanation. All right, so we have a researcher that's studying the relationship between pattern recognition receptors present within host cells and Crohn's disease. The pattern recognition receptor nucleotide binding oligomerization domain protein 2, NOD2, has been shown to detect conserved motifs in bacterial peptidoglycans such as intracellular muramyl dipeptide and promote activation of host inflammatory pathways. So let me stop right there, because this is a situation where you want to stick to the things you know, because you're going to see things that are kind of thrown at you in questions that, are different than anything you've seen in your study material. So, you want to stick to the things you know. So what do we know? We know a little bit about pattern recognition receptors, which I'll talk about in a minute. Okay, and you might have heard of NOD2. If you haven't, that's okay. We'll talk about that. The bottom line is, they're basically saying here that the pattern recognition receptors, one example of the pattern recognition receptors is NOD2, right? So we can kind of write that down. So pattern recognition receptors, and we'll talk about these in a second, they're basically telling us that an example of a pattern recognition receptor is NOD2, okay? And NOD2 is going to detect conserved motifs in bacterial peptidoglycans, and they give you an example here, and promote activation of host pro-inflammatory pathways. Even if you didn't read that, right, you should know, right, from the video series we've kind of just covered in this chapter, what pattern recognition receptors are and what they do, right? So pattern recognition receptors are going to recognize pathogen-associated molecular patterns, or PAMPs, right? They can also recognize damage-associated molecular patterns, right? So these receptors can kind of recognize these very non-specific patterns in certain bacteria or viruses, for example, right? And we kind of talked about some of these. So we said, look, there's some PAMPs, for example, LPS, right, and gram-negative bacteria. That's very non-specific. A lot of different gram-negative bacteria, right, they're going to have LPS, and we can recognize LPS non-specifically. It's not a specific antigen and a specific idiotype that are coming together. This is just a, LPS in general sets off this host inflammatory cascade. It's very non-specific, right? So LPS can do this. We said viral nucleic acids can do this. Even bacterial flagellin is another classic example. And then the damage-associated molecular patterns are usually things from host cells. So, if a host cell is, has defective DNA repair or something along those lines, for example, there's going to be very non-specific patterns that can be recognized to target that host cell for destruction. Again, these are non-specific. So if something is non-specific, it's generally going to be part of the innate immune system, right? So this whole process is really part of the innate immune system, because again, these are non-specific patterns that kind of set off that initial cascade. When you have a pattern recognition receptor like NOD2 or a toll-like receptor, which we kind of talked about, TLR, toll-like receptor, these can recognize these non-specific patterns. And what they end up doing is once these bind together, right, once LPS binds to this toll-like receptor, or flagellin binds to, something like NOD2, whatever it is, what ends up happening here is we eventually end up getting an inflammatory cascade. Okay, and this is kind of what we talked about in the last video. And the way this all starts is by increasing transcription of NF-κB, which is particularly high yield. Okay, we talked about this being a very important step in starting this inflammatory cascade. And then this kind of sets off multiple cytokines like interleukin-1 to change temperature set point, interleukin-6, right, TNF-alpha, all of these kind of acute phase inflammatory reactants. Okay, and so this is kind of how this whole process works, and that's what you're kind of expected to know. You're expected to know this is very nonspecific, sets off an inflammatory cascade through the regulation of NF-κB, and then kind of the big ones to remember here are TNF-alpha, interleukin-6, interleukin-1, maybe even some interleukin-8, right, for chemotaxis. With all that being said, going back into the question here, we're going to read this line again. The pattern recognition receptor NOD2 has been shown to detect conserved motifs in bacterial peptidoglycans, where you just said, we already know that, right? Conserved motifs, right? Things like flagellin, LPS, etc., right? That's something we already know. And that gives you an example of one here, intracellular muramyl dipeptide. And when doing this, it promotes activation of host pro-inflammatory pathways. Okay, that's very important. So NOD2 has been known to be highly expressed in the Paneth cells of the intestine. That might have something to do with the fact that they're looking at this relationship in Crohn's disease, right? The researcher attempts to induce mutations to further elucidate the effects of ileal inflammation on NOD2 deficient mice. So we want to see the effects of inflammation on NOD2 deficient mice. Key word here is deficient, right? So these mice do not have this, presumably this gene that codes for this NOD2 receptor, right? So based on the information above, which of the following would most likely be impaired in NOD2 deficient mice? Okay, what would most likely be impaired? Now, if you kind of understand this, which is what we kind of talked about, it should make sense that NF-κB activity would actually be impaired. And you could say, well, why is that? Well, remember, these mice don't have this NOD2 receptor, right? So if they don't have this NOD2 receptor, we can make the assumption that, look, if I don't have this receptor, if I'm deficient in it or it's dysfunctioning, whatever it is, I'm going to have an issue setting off an inflammatory cascade, right? I'm going to have an issue setting off an inflammatory cascade. So what will happen is potentially, right, I'm not able to go through this next step to upregulate NF-κB because I don't have the receptor to make that whole process happen. I don't have the receptor that then binds to, in this case, this intracellular muramyl dipeptide, right, to set off this inflammatory cascade. I don't have the receptor to bind to flagellin or viral nucleic acids or LPS to kind of go through these steps, right? So if I don't have that receptor, then everything downstream is affected. So I might have, low levels of interleukin-1 in the setting of an infection, or interleukin-6 or TNF-alpha. In this case, it has to do more with Crohn's disease, but the point is still the same. The point is that we can't set off the inflammatory cascade, and the point is that you should understand that these pattern recognition receptors are associated with the innate immune system and setting off the inflammatory cascade through the regulation of NF-κB. Now let's quickly just go through some of these other options here. And if you're just kind of watching these videos sequentially, we haven't really talked about some of these other things yet, but I'll just give you a little bit of a rundown as to why they're not correct. So B7 protein binding to CD28. So B7 and CD28 are kind of linked because these two come together essentially to perform a co-stimulatory signal. And this has to do with the activation of T cells, which ends up being part of the adaptive immune system. And this is done in situations where you have a specific antigen, right? And you might be creating a memory response, for example. In these situations, though, again, this is going to be more adaptive immune system, not really directly related to the NOD2 receptor or this innate immune pathway that we're talking about. Okay, so not directly related to this and would be unlikely to be directly affected. Certainly not the best choice here. CD40 membrane receptor activity. Now, this is actually a pretty big one. So CD40 and CD40 ligand, which we're going to talk a lot about in this series, kind of come together to allow for class switching of antibodies. Now, remember what we kind of talked about. We said, look, IgM is kind of like this generic antibody that we have. And we can make more specific forms of our antibodies by adjusting the isotype. Okay, so we can adjust the isotype of the antibody to make things like IgG, right, great for our memory responses, IgA at mucosal surfaces, IgE, right, etc. So we can make these different types of antibodies, but to do this, to undergo this process, we have to undergo class switching. Okay, so we have to do some kind of class switching to let this happen, to change the isotype. And to do that, the key receptor binding to do that is going to be the CD40 ligand, which is going to kind of set off this cascade. And I'll go through the entire pathway, all the details, as we kind of progress through this unit. But what I just want you to understand here too, since we're on the topic of this, I don't want to go too far down into the details yet. So you have a heavy chain, which I'm drawing in black here, and then you also have a light chain. Okay, so you have a heavy chain, and then you have a light chain. And the heavy chain has kind of two regions here. So this pink region is the constant region, okay? So we just say this is the FC region, the constant region. And then we also have a FAB region, an FAB region. So the light chain and this part of the heavy chain are going to make up the FAB region, or the FAB region. When you're changing the isotype, right, this is what we're doing to go from like IgM to IgG, right? When you're changing the isotype, you're going to be changing the FC region here, okay? When you're changing the isotype. Now, if you changed the idiotype or changed the variable region where this antibody can bind, that's going to be in this area, in this FAB region, okay? So this FAB region is going to contain, hypervariable regions that bind to very specific antigens that underwent VDJ recombination, whereas the FC portion is going to be more regulated for complement binding and the isotype. Anyway, the point of this is because IgM is kind of like our generic antibody, IgM can be thought of as part of the innate immune system, right? Because it's kind of our generic antibody. But for us to go to class switching, that's where we're now kind of transitioning into the adaptive immune system, right? Because we're creating antibodies for memory responses, making antibodies like IgG, for example. And so we can say that IgG is actually more part of the adaptive immune response because we require class switching. And again, we're primarily talking in this question about the innate immune system specifically, right, with the NOD2 receptor. The class switching would be kind of out of the scope of what's being tested, in this study. All right, so if we look at option D, this is really just along the same lines of option C. It sounds kind of complicated, alterations within the heavy chain constant domain of immunoglobulins. What does that mean? It's just what we just said, right? Here's the heavy chain, right? This is the heavy chain, this black line. Here's the constant domain. If I change the constant domain, I end up with IgG, IgA, IgE, right? Some different form of the immunoglobulin. So this would not be the correct answer also for the same reasons, because it would have to do with class switching, adaptive immune response. Now the last option on here, terminal deoxynucleotide transferase activity. Now what this is, and you do have to know a little bit about this, but you don't have to know a ton of details, but what this is it's a DNA polymerase that kind of randomly adds nucleotides to immature T and B cells. Now the thing about terminal deoxynucleotide transferase, number one, you sometimes kind of think about this when you're thinking about things like RAG1 and the RAG2 genes, which are basically like recombinase genes that allow for VDJ recombination to create this kind of hypervariable region here to make all different forms of these, idiotypes, for example. Now, this process, is not directly related in any way to what we're talking about with the pattern recognition receptor, so it's clearly not the best answer. But the other thing I want you to understand when you see TDT, right? If you see TDT positive, in a question stem or a lab value, you start to think about, a little bit about, especially in the setting of blastic tissue, you start to think about acute lymphoblastic leukemia. And one of the ways that we start to differentiate the different forms of leukemia is you can look at acute myelogenous leukemia. Typically, AML is TDT negative. And that's because the blastic cells in AML typically are from the myeloid lineage, not the lymphoid lineage, right? The lymphoid lineage would typically have more cells that might have this TDT, this polymerase, essentially, that's allowing for VDJ recombination in these very immature blastic cells, right? Blastic cells are immature. So the point is that you can see TDT positivity in ALL, especially in the setting of blastic tissue. That's what I want you to remember. But again, this is not directly related or linked to this question, right? If you understand this pathway that we talked about in the previous videos in this chapter, you can pretty much solve these questions where they start to throw some information at you that seems a little convoluted, seems a little complicated. Just stick to what you know. Look for the things that you know and stick to the pathways that you've kind of gone through already, and usually you can decipher some of this. Hey everybody, Dr. Reviso here. In this video, we're going to talk about inflammation, and this is a really big section overall, but what I want to do is I really want to focus on what are the major things that we have to know in this section before we move on. And this section's categorized, differently in different textbooks and that kind of thing, but we're going to cover a lot of the components surrounding inflammation.